Welcome everyone, and thank you for joining us. My name is Paul Hill, and I'm delighted today to have special guest Stephen Stamp, the CEO and CFO of Midatech, with me, a leading drug delivery company specializing in rare and serious cancers. Welcome, Stephen.
Thank you, Paul, and good afternoon, everybody. I'd like to begin the presentation today by giving a short overview, if I may, of Midatech. I'll start with the disclaimer and just, if I may, just take that as read for today's purposes. So Midatech is a biotech company, as Paul has intimated, focused on drug delivery, where we have three proprietary micro and nano technologies, and more of that in a second. We pride ourselves on our speed of innovation, and I can tell you about some of the things we've been working on. The company is dual listed on AIM market in London and NASDAQ in the US, and our headquarters, following scaling back of operations announced at the end of March, is based in Cardiff, where we have R&D capability, a non-GMP pilot scale manufacturing plant, and our administration offices. Today we have 20 employees. So I mentioned three technologies. They are, the first of which is Qsphera. This is a technology focused on sustained delivery of drug over time using a patented PLGA-based formulation, which again I'll discuss in a second. The second is Midasolve. This is based around the local delivery or targeting of drug, and we have a lead compound in phase 2 there. And the third is Midacore, which is a gold nanoparticle-based technology that's focused around targeting both therapeutic agents and also targeting agents. The common theme across all three of these technologies is that they are focused on improving the biodelivery and biodistribution of pharmaceutical agents. That's both drugs that are currently on the market, and we're also beginning to work on the proprietary APIs of other pharmaceutical companies. So let's start with Qsphera. This is a technology based around the microencapsulation of PLGA. That is polylactic co-glycolic acid. It is a biodegradable and biocompatible substance that is quite commonly used in foodstuffs and in pharmaceuticals as a carrier of drug. What we do is we combine PLGA with active ingredients in microspheres, and these can then be injected into the body to form a depot. And that depot then releases drug over an extended period of time, could be weeks, could be up to 6 months. What makes our particular PLGA technology different is that we use piezo printing. So how does it work? Well, the drug, if you look at the diagram on the right-hand side, the drug is mixed with a solvent, and then the solvent is then squirted through a printer. And while the droplets are in the air, they are then subsumed into a vortex of anti-solvent, and out pops the other end millions of these microspheres, which has the drug encapsulated every second. So it's a very neat technology. It's very small footprint and eminently scalable, unlike some of the common currently used methods for PLGA manufacture, and more of that in a second. One thing we're able to achieve here is incredible monodispersal, and by that I mean there is very little variability in the size, shape, or drug concentration in our microspheres. What you see on the current slide 8 is a picture of a Qsphera microsphere compared with one of the competitor products called Sandostatin LAR. This is a product that's been on the market for a number of years by Novartis. And you can see the homogeneous size of the Qsphera product compared with the rather random variation in the Sandostatin product. we're able to encapsulate proteins of up to 50 amino acids each. Excuse me, I should have said peptides. And now we're beginning to work on proteins such as monoclonal antibodies and Fabs. We're evaluating those on a case-by-case basis. We can get up to 25% drug loading versus polymer in our microspheres. And overall, it's a vastly superior methodology with a lower cost of goods than currently available therapies. What does that mean clinically? Well, on this slide, what you see is some quite complex charts, but they relate to a product that we developed called MTD-201. This is our version of Novartis's Sandostatin, the product I referenced earlier. So if you look at the right-hand side of this chart, there are three graphs. The top left graph illustrates the blood levels by individual patient of octreotide, which is the active ingredient, over a period of time. And you can see that the shape and profile of the blood plasma levels is remarkably consistent between patients. If you look at the top right-hand chart, this is the same measurement, but this time for Sandostatin LAR. And you can see that the interpatient variability is very significantly different with that product. And if you average out those products, average out those charts by person, I'm looking at the third chart on the right, you can see that the Novartis product, which is the orange chart, produces a very significant burst release followed by a long tail on average, whereas the Sandostatin product is much more consistent over time, and that's the blue line. Moving to the left-hand side of this chart, one of the other things that makes our technology unique is that we're able to deliver the product subcutaneously and not just via intramuscular injection. This confers a number of advantages in that patients would be able to administer the product themselves at home, which means less outpatient visits, less cost of the system, and therefore attractive from a payer perspective. And because the formulation is less viscous than the currently available therapies, we can use a smaller needle gauge, which means less site irritation, greater injectability, and less pain for the patient as a whole. So overall, a better product. This product is ready to go into phase 3. We weren't able to fund that ourselves, but this product remains available for licensing to third parties. One of the more interesting things that we're now working on down here in Cardiff is the formulation of a long-acting protein, specifically monoclonal antibodies. Nobody has been able to crack this as of today, partly because proteins are quite fragile and they can be easily denatured during a complex manufacturing process. We think we might have an opportunity to formulate a protein using Microspheres technology for a number of reasons. One of which is that our formulation does not have large numbers of voids in the microsphere, and voids are not helpful because some of the solvents that are used in the process can get into the void and denature the protein. Secondly, the solvents we use are less toxic, in fact non-toxic, compared with the traditional manufacturing methods, and we don't use significant shear forces or heat. So we think there's quite an opportunity here for us to be able to formulate monoclonal antibodies, which are typically given by injection or infusion. This would be a much more convenient and less costly way of administering those types of drugs. I can't say we've cracked it yet, but there are useful signs that we're seeing that we think we might have a solution for that in the coming months. Moving on to our second technology, Midasolve. So this is the solubilizing of inherently insoluble molecules. And what we're able to do is to convert drugs that are only administered orally, because they're insoluble, into drugs that can be administered via injection or liquid. The advantage of that is that the drug can then be targeted on, for example, a tumor. which you couldn't do if you took the drug orally. A lead compound using the Midasolve technology is called MTX110. This compound is a combination of our formulation of panabinostat combined with a CED system. That stands for Convection Enhanced Delivery. So what we have done is formulated panabinostat using Midasolve to make it soluble. The product is then administered via a shunt under slight pressure, that's the CED, directly into the tumor, specifically brain cancer. Panabinostat is an approved product. It's on the market for multiple myeloma, but suffers significant toxicity issues when administered at a high dose. We're able to deliver about 100,000 times the concentration that you would normally see with an oral dose into the brain tumor using our system without any apparent systemic toxicity. The first indication we're looking at here is called DIPG, which is diffuse intrinsic pons glioma. It's an ultra-orphan cancer of the brain found in children. About 1,000 patients worldwide are diagnosed each year with the median survival time of about 9 months. There is no useful therapy for this indication currently. Radiation is sometimes used, but it can have deleterious effects on the brain. We have an ongoing phase 1 study with the University of California in San Francisco to assess safety and tolerability. We expect to be able to report on that study quite shortly. Thereupon, we will move into a phase 2 study with Kinderspital Zurich and Princess Maxima in Utrecht, two of the leading pediatric cancer centers in Europe. Whereupon, if we can keep 12 of the 19 patients alive after 12 months, we would expect to apply for early approval of this drug based on first-in-class and no alternative therapies. We are at the same time conducting an exploratory study in Columbia University using an alternative CED system to the one that's being used in California. And we're also doing an exploratory study with the University of Texas in medulla blastoma, which is kind of an adult form of DIPG. If either or both of those are successful, we would then consider taking the product into a larger market called glioblastoma multiforme, which is probably a $3 to $5 billion market. Again, with no, there are therapies, but none of them are particularly effective, and the cancer tends to recur. That would be a home run for this product. We would expect to have data from the UCSF study very shortly. The phase 2 study will finish sometime in the second half of 2021. And then depending on how those look, we will move into GPM shortly after that. The third of our technology is Mitacore. This is the gold nanoparticle technology. It's probably about the smallest therapeutic molecules around at 2 to 4 nanometers. And we're able to deliver drug on a very targeted basis. We've got some preclinical data using methotrexate to treat psoriasis in a mouse xenotransplant model. What these 3 pictures on the right-hand side show is the impact of Vaseline, which is as good as placebo, on skin irritation and inflammation. The middle picture is the current standard of care, which is Divabet, and you can see it's better than placebo, but still quite a lot of inflammation and irritation. And the right-hand picture is our gold nanoparticle product, which is much improved. Now, this is only a xenotransplant mouse model, hasn't been translated into humans yet, but we are discussing the potential partnership of this product with third parties who take it into the clinic. Which brings me to our strategy. On March 31st, we announced that we were going to no longer fund MTD-201, the octreotide product, in-house. it was just getting too expensive to take the product into phase 3. We would have needed about $30 million, of which the Spanish government would have contributed about 8.5 or 9. Given the COVID situation and the markets, we didn't think we could raise that money, so we put that product on ice. We shut down our Spanish pilot manufacturing facility, and we more or less cut the burn rate in half. Our new sort of revised slim down strategy is to work on 2 strands, one of which is to develop an in-house portfolio of currently marketed active pharmaceutical ingredients. And there are 2 of those we're currently working on. It will take about 3 months to get to an optimization on proof of concept, at which point we could take the product into animal models. We do not intend to engage in human clinical studies. We would look to partner either or both of those products before we did that. And at the same time, we're also beginning to work on a fee-for-service basis with third parties to work on their proprietary APIs. And you may have seen a few days ago, we announced the first of those with Dr. Reddy's. There's a first molecule we're working on. There are others behind that. And we have 1 or 2 other agreements that are currently in discussion, which I'm hoping to be able to announce in the coming weeks. So this will produce revenues, cash flow, and validation of our system. And this is in addition to MTX110, which we continue to develop from our own resources. Just one slide on the financials for last year. Our revenues are very small, and about half of that was milestones from third parties. The other half was grant monies. We spent about £8 million on R&D. That will be materially cut this year because of the cessation of MTD 201, and just under £4 million on administrative expenses, again, because of the reduction in force that we effected in March and April. That you could also expect that to be smaller this year. Cash at 31st of December was £10.9 million. However, that was offset by about 5 million of loans that we had from the Spanish government, which we had to repay. And there were also another million or so of grants on top of that. So those have been now repaid. But in addition, we've also raised 3.7 million net a couple of 3 weeks ago. So our current cash runway takes us into the second quarter of 2021, which gives us a decent runway to effect something which would be maybe a sale of the company, could be a sale of MTX110 or a sale of Microspheres or both, and then the sale of the cash shell before that cash runway runs out. So just to summarize then, we have 3 quite innovative drug delivery platforms, all of which are being clinically validated. They have multiple applications. We have a very large IP portfolio with 36 patent families and over 100 issued patents. Our licensing strategy is beginning to pay off, as the Dr. Reddy agreement illustrates. And our burn rate has been cut to about 400,000 a month, which is just less than half than it was a couple of months ago. So that was all a little rushed, but I'm happy to take questions as you wish now. Thank you.
Thank you very much, Stephen. If anybody would like to ask questions, there's a box hopefully on your screen you'll be able to put through. Just kicking off, Stephen, and I guess it's probably is the $64 million question. And I know you are legally obliged that you can't talk a great deal about it, but how is the formal sale process progressing?
So we went far and wide to gauge interest. We employed a boutique healthcare investment bank out of Florida called Noble Capital Markets to help us. I've worked with them before in their former guise as JSP partners. They're very experienced. They have a fantastic network. And we have a number of parties who, who have engaged at multiple levels. Some have expressed an interest in MTX-110. They might be a cancer or an orphan company. Some have expressed an interest in the Microspheres drug delivery platform. They might be drug delivery companies or companies that have suitable APIs to which it could be applied, or technology companies. And then a few people have expressed an interest in the whole company, either as an acquisition or as a reverse takeover candidate. So we have quite a good mix in there. I can't tell you that any of them are at term sheet stage, because if they were, I'd probably be making an announcement. But a number of them are active and doing due diligence.
Great. And what's the sort of timeline indicatively that you would hope this process completes? I'm guessing sort of 3 to 4 months after April. Would that be reasonable, or is that
Probably a
ambitious?
little bit longer than that. We've set ourselves an internal target as the second half of this year, but I think realistically that what that really means is before Thanksgiving, because after that the United States starts to wind down for that, and then Christmas, and then you can't get anything done until after the new year, by which time our cash runway becomes precariously short again.
Okay,
So
good.
I think we'd be looking to do something pre-Thanksgiving.
Okay, and then you're in the lucky position of having the second $64 million question, which is to do with the letter that the company received from Sakura. Can you just give us a bit of background in terms of, given it's a non-exclusive license, what's the rationale on their side to do something like this? It just seems, given the potential opportunity for them to get royalties, it seems very strange behaviour.
It does. So we're currently in a short period, which the agreement provides for, during which we are able to carry out good faith negotiations. Those haven't resulted in anything yet, and I can't claim that they will, but I don't want to prejudice that by sort of speculating on what Sakura Bio's motives may or may not be at this stage. You're right, the license is non-exclusive. So if they wanted a second party to develop a product like MTX 110, in theory they could, although we have our own IP which protects our product. So maybe they have an interest in that. Time will tell. And when we know the answer to that, you can probably expect to see a further announcement.
Okay, and does that this sort of like uncertainty with the MTX 110, does that, and their letter coming in, does that sort of like impact at all the sale process? I mean, the attractiveness of the asset.
It does. There's no doubt about that, Paul. It certainly creates some uncertainty. I have to tell you, though, that it's not all bad. The terms of that license are not particularly favorable to Midatech. However, we wouldn't be able to access the market with panobinostat on the timelines that we currently have without some kind of license to that product. So I'm hoping there's going to be some middle ground where we can make peace and move on. If not, then I think it is reasonable to assume that the overhang from Sakura Bio's actions will impact the value that we're likely to achieve for MTX 110. Yes.
Okay, well, I mean, the good news is, I guess, is that we've got an organic growth strategy anyway with the other products. But just with that lead candidate, the MTX 110, can you just talk a bit about how much, what's sort of the advantage, what beneficial advantage it has over existing sort of standard of care? Because a lot of investors have read a great deal about immunotherapies with Merck's Keytruda, and likewise with Bristol Myers is Optiva, and then also sort of proton beam, and the radiation side, and also Accuray's CyberKnife. It's all in the NHS. How much better is this potential lead candidate over those existing technologies?
Well, I'll do my best. Please understand that I'm not a medic, and I'm certainly not an oncologist. But I'm losing my voice here. DIPG is a tumor that grows right at the base of the brain, the top of the spinal cord. It is not operable because of its location, and oral drugs don't tend to be effective because they don't generally cross the so-called blood-brain barrier. Mother Nature put that blood-brain barrier in place to stop toxins getting into the brain for a good reason, and it's pretty effective. So there are no current therapeutics for DIPG used at all. The only thing I believe that is occasionally used is radiation, and that has, unless you're highly skilled and very focused, you're more likely to do damage to good brain cells, or as likely to do damage to good brain cells as you are tumor brain cells, and therefore it carries its own risk. What we've been able to do is take a drug panabinostat, which we've shown in a lab in vitro testing against a panel of about 80 other chemo agents against human DIPG cell lines, that it is the most effective at killing those tumor cells. So we took that drug, we formulated it, and we then passed the drug down a catheter directly into the tumor of the child, in the child's brain. So we're crossing the blood-brain barrier and we're getting right into the tumor. And the more sophisticated versions of these catheters actually are able to direct the drug, left, right, up, and down. So you can effectively spray the drug around the tumor until you think you've killed it all. And you repeat this process again and again. So it is not a easy drug to administer, but it's the only thing that we know of that's got a chance of working today on the basis there are no current therapies.
Okay. And then if it all goes well in phase 2 trials and we get a result the second half of 2021, what would then be the next steps for the company? I presume you would license it out to a large biotech company that could then take it through phase 3?
So that's a fantastic question. Because this is ultra orphan and because there are no other currently available therapies, we would actually seek an early approval for that drug after phase 2. I
Oh,
can't
wow.
guarantee that we would get it, but we think there is a reasonable prospect that we get an early approval, maybe with some post-approval monitoring obligations in terms of survival rates and adverse events, which is not unexpected and quite appropriate. So that would be our best case.
And if you got
If we
that
can't
best
do that, and it
case,
does, sorry. Mm-hmm.
if you got that best
Go ahead.
case, then would you actually commercialize it yourself? I'm guessing it'd be an extremely valuable asset to have, but I'm guessing you haven't got the footprint with the relevant in America or across Europe or Japan to actually do the internal selling yourself. You'd then look towards somebody who's into cancer for pediatrics.
So that we would evaluate closer to the time. And the answer to your question is probably yes, although I'm given to understand that because of the ultra rare nature of this condition, the patients tend to be treated in relatively few centers around the world. Like 3 or 4 in the US, 3 or 4 in Europe, 2 in Japan, Korea, and 1 in China. I'm making those numbers up, but it really is a relatively small number of centers. And you can imagine that the surgery required here requires a certain amount of skill and training. So you'd actually want the patients concentrated in these centers. The patient support groups are very active, so it's not hard to find the patients and to find the centers. So I'm actually would be intrigued to, when we get to that stage, to work out how many, there would be a bit more than reps, but sort of medical science liaison types of individuals we would require to be able to promote and market this product ourselves. I'm guessing it'd be less than a dozen. So if
Right.
that were the case, then we might take it on ourselves. But as I say, we'll evaluate that nearer the time.
Okay. And given that the company's now downsized its development and R&D capability, and it's got 20 employees, I'm guessing the sort of resource availability internally is still sufficient to be able to get through this phase 2 trials and any other sort of like development opportunities over the next 18 months.
Yeah, so we have two of those 20 are, I'll tell you how they break out. We've got three in administration, including me. We've got two in clinical ops, and the rest are our lab folks working on the Microspheres formulations, and they're currently working on 5 in parallel.
Okay.
So the two clin ops people are working pretty hard, as you would imagine, and also we use external vendors as necessary to fill in the gaps.
Okay, that's good. And then just in terms of COVID-19, it's obviously been well publicized across the industry that anything non-COVID related in clinical trials has been delayed. How you is that is impacting any of the work at all? I did notice in one of your I think it was in the prelims yesterday that the company mentioned one of the actual compounds that still needs another patient to complete its the total number of people we need to test in phase 1 out in California. So have you got any color at all? Any further information in terms of the actually executing the clinical trials given COVID?
Yes, that's a very good question. Because of the lockdown, particularly well here and in the US, patients haven't been able to travel to these centers
Hmm.
to get treatment. So that has slowed down recruitment in Columbia, New York, Texas, where we have another trial, and San Francisco. In fact, we now have the last patients in San Francisco. We're just waiting for that patient to finish treatment, and then we'll be able to close that study. But overall, yes, it has slowed down recruitment.
And how many people are actually in that San Francisco study? Is it, given it's phase 1, is it sort of 10 to 20?
7.
7, okay. And you're testing safety as well as efficiency.
Efficacy. So actually, there are two: safety and tolerability. So the study design has a dose escalation in it, and what we're
Yeah,
trying to do is to assess the maximum tolerated dose for MTX 110 that we could then take into the phase 2 study. Then
yeah.
the second thing is to record adverse events, or particularly drug-related adverse events. Yes.
Okay. And then I guess in the phase 2 study for the MTX 110, which the results have been 18 months, it's both the efficacy and the safety, given that it's an orphan drug status, and you would ideally, your ideal scenario is that you're going to be able to then begin potentially commercialize or seek approval to commercialize it after phase 2.
That's correct. The primary endpoint for that study will be survival, and I know it's gruesome to talk about these things in the context of these patients, but that's the reality. And the hurdle will be having 12 of 19 patients alive at the end of 12 months treatment.
Okay. And what's the sort of the market opportunity for the Q-Sphera, the drug extension product? As in, like, is there multiple opportunities there when we're looking at a few at the moment in sort of cancer? How do you see that? Because it seems to be, should we say, a bit of science that could be applicable to a lot of areas in medical, verticals. Is it got a lot? Is it got a very wide sort of applicability?
It does. It does. There are some limitations based around, for example, dose. If the drug is a very high dose, there's only so much that you could load into a depot without it creating a bulge in your abdomen, if you could imagine.
Mm.
So that's one limitation. There are other limitations around solubility and compatibility with PLGA. But the main limitation, I would say, is you need to have a benefit. You need to create a benefit for the patient which is quite easy. It's often around convenience. Having one injection every 3 months is better than taking several tablets a day every day. But you also have to have a benefit for the payer system. Otherwise, they won't reimburse for the drug. So, to give you an example, whilst it may be more convenient for a patient to have one injection every 3 months, if there is a generic tablet at a penny each, the payer will insist that the patient takes the tablets every day rather than will reimburse for an injection. So you need to have
Okay.
um, both, and the types of drugs, for example, that would benefit most from Cusfera are those which may have a narrow therapeutic window, for example, where you're trying to deliver nice, consistent, even blood plasma levels, like the chart I showed earlier. Whereas to venture outside that therapeutic window with too low a dose, which you might get if you miss a dose, we get breakthrough issues, or you get too high a dose, in which you get toxicity, and where the breakthrough issues cause much greater costs to the healthcare system. So, for example, and a particular example, there is a product on the market, risperidone, which is an antipsychotic product. You can imagine that if the patient doesn't take his product for whatever reason, the cost of the system as a result of that could be extremely high. And therefore, there is an advantage to a product like Cusfera long-acting depot.
Okay. And then just switching gears on the MTD 201, is there any progress at all on seeking potential partners for that particular drug, or is that sort of stay where it is at the moment?
So we're not going to invest in it anymore. We do have a few ongoing discussions about MTD 201. I can't promise you that they are going to yield anything positive, but there are people doing due diligence and looking at it. Yes.
Okay, great. And then just in terms of the actual listing, given we're a sort of £10 million market cap businesses, having two listings, one on in America and one in the UK, does that make sense given the cost base?
It is for sure more expensive, and the US is more expensive than the UK. I think had we not had that US listing, we wouldn't have been able to do the last two fundraisers, and the
Okay.
company
Yep.
would have been in considerable trouble before now. What I've generally
Okay.
found is that the London market, when it's open, it's open, but when it's closed, it's closed. Whereas the US, you can generally raise money there. It may not be on the best terms, but the pools of capital are so deep. You're usually able to raise money at a price.
Okay. And then just sort of like, we're running out a bit out of time, but sort of like just ending on a positive in terms of the sort of the news flow going forward. So there's a lot of investors obviously very interested in the technology. How do you see the news flow over the next sort of 2 to 3 months, etc.? Which sort of like key milestones for people to look out for?
I think you might see another partnership, a bit like the Dr. Reddy partnership, but maybe this time focused around a protein or monoclonal antibody with a big pharma. You might see the result of our formal sale process, which could be a sale of one or more of the assets or the whole company. You'll see in the next probably 6 or 8 weeks the data from the UCSF study on NTX 110.
Well, great. I mean, that's got three positive things potentially coming through on the pipe over the next few months or so over the summer. So, well, thank you very much, Stephen. It's I've been absolutely fascinated by the science. I'm not a sort of like yourself a cancer expert, that's for sure. And there's certainly some very exciting drug candidates you've got there. So I really appreciate your time taking us through the company's presentation. And of course, to the wider audience, I thank very much for your questions, which I've tried to answer, we've tried to get answered, and likewise for joining the webinar. So thanks very much, Stephen.
Thank you. Thanks, everybody.
And look forward to hearing everything going forward.
Okay, thanks a lot.